So we are going now to start after this general introduction from Agnès and Clarisse, really to talk more about diatoms and phytoplankton. So today it will be this afternoon it will be diatoms, and tomorrow morning again diatoms. That's where we have more experience with Agnès and also Kalman, who is in the back there. So Kalman, maybe when we need your help, we will make you a sign, and if you can help us, please. Okay, great, thank you. So really this first part will be some reminders, at least for some of you. And what I can propose you, it will be half an hour talk. Some reminders about what are diatoms, so basic things about diatom biology. I wanted also to show you the diversity that we can have with diatoms, so some notions about diatom taxonomy. Just for those who don't really diatoms, it's not the case of Milos, of course, he knows diatoms for sure. And also for people who don't know really what is biomonitoring, I will give a very brief talk about classical diatom biomonitoring. Are you okay with that? Okay. So let's start with diatom biology. Just a few words about their diversity and importance in the biosphere. There was this estimation, which was carried out by David Mann and Piet van Ormelingen in 2013, and they estimated the diversity of species of diatoms to more than 100,000 species. That's enormous. That's crazy to see that's such a diversity. So of course, when you consider the diversity, which is mostly known in lakes, in fresh waters, it seems to be not thinkable to have such a big number. But when you consider the number of environments which have not been, where no diatomids have seen any samples, for instance, when you go in tropical areas in Africa, there are many places where we don't know the diversity. Also, when you go in marine environments, probably not in the plankton, in the marine phytoplankton, but as soon as you take bottles and a mask and you dive a little bit in 10 meters, 20 meters, and you start to brush the biofilm, the diversity of diatoms is again quite unknown. And this is how they estimated the number of species, considering the number of habitats which are unknown for diatom diversity. 25% of the total biomass on the Earth, and yes, they colonize all habitats. So of course we think about rivers, lakes, oceans, but also we can find diatoms in soils, in wet walls and caves also. If you have some light, of course, you have a lot of cyanobacteria. There are some people working on cyanobacteria here, but also you have some diatoms which are living in this kind of very extreme environments. Yes, I put a few photos because I like looking at diatoms. There are some people who like to do yeah, Daniela, of course, yeah. Okay, a few words about the cell organization. This is a scheme which was drawn by that you can see. Many of you working on diatoms, you know this scheme of Ronman and Crawford. Sorry, there is a mistake here in the book that everybody knows if you're working on diatoms. So diatoms, they have an extracellular skeleton, which is in siliceous, which is made of an epivalve and an hypovalve that you can see here on these two photos. Here on the bottom photo, it's a living diatoms. Both photos are the same species. It's Melosira varians. Here it's living. So you can see all the organoids. You can see small plates here. These brown plates are the chloroplast. So there are many inside one cell. Here you have one cell. Here a second cell, and it's making yes, not some ribbons, but some filaments. All these are lipidic drops. And here you can see the diatom when it's dead. You can see only the frustule, and you can find again the epivalve and the hypovalve. And so these two parts of a frustule are connected with what we call the gridal bands. Yes, it's really sticking these two parts in one. And some diatoms, they have this long longitudinal fissure here that you can see here. which enable diatoms to move. And they don't have all this ability to move. A few words about the life cycle. Diatoms, they can reproduce in two ways. The first one, it's a vegetative reproduction by simple mitosis. So this is a cross section of a diatom. So you can see here it's the cell, and you can recognize here the hypovalve, the epivalve, so this extracellular frustule. And if you have a cell division, a simple mitosis, so you will have two daughter cells here, and each daughter cell will keep with it one part of the mother skeleton, and it will produce again it's a new skeleton, but which will be always smaller than the mother one. And so if you have like that several mitoses along the days, days after days, in fact, you will have a cell size reduction from the mother to the daughter. That's what you can see here. This is a paper of Elaine Cox and Rose, one of her PhD students. What they did, but it's something which is quite well known since a long time. But in fact, they studied a particular species which is called Gonphonema parvulum, a very common species in rivers. So they put it in culture, and day after days, they can see that there is a mitosis and this cell size reduction. So this is the same species. Here you have the mother cell, and here the youngest cell, let's say. The second kind of reproduction is sexual reproduction. So here again, you can find this this nice scheme in the book of Ron, Crawford and Mann. So if you have here a vegetative cell, you will have progressively some mitosis. So this is the classical reproduction that I showed you just before. So you can see here there is a cell size reduction. And if at one moment the environment is stressful, this cell will go into meiosis. So from two N chromosome, it will produce some gametes with N chromosome. That's what is symbolized here. And these gametes will mate each other. So that's what we call a sexual reproduction. Will produce an oxospore here. It's in fact a resting spore. And these spores, in fact, they are able to resist if you have a bad condition in their environment. And we can find, for instance, this kind of species, you can find it in lakes. And during some bad period for this kind of species, they are forming this resting spore, they are falling into the sediment. And when the environment start to be again interesting, so this resting spore is making a mitosis, and you will find again the largest initial cell. So now let's have a look to the cell organization. Here you have a photo of a culture that we have in a culture collection. So here this is a Pinularia, a species of Pinularia. Here it's only the frustule, and this is the living cell here. And here you have a scheme of what we can see here. So what we can see here is two chloroplasts. So here they are gray, but they should be brown. So you have two chloroplasts here. The position of the nucleus is here. So that's what you can see here. And you have sometimes you can find here these lipid drops. So you're going to see a little bit what Clarisse showed us. We're going to enter more into the details and the genetic material that we can find in each organelles of the cell. So if we consider the nucleus, we have several chromosomes in the nucleus, and they have coding genes for the ribosomal RNA. And so RNA, as you know, this is what is symbolized here, is giving the ribosome after that. And we have So in this ribosome, you have two subunits, the large subunit that you can see it here. So it's 16S, which is composed of several genes. So we have 28S, 5.8S, and 5S. And the small subunit here, which is composed of 18S. And so the 18S is what so Clarisse told you is just before. It's a marker which is widely used in biodiversity studies because yes, you have variable regions. You remember this graph with the entropy. You have some variable regions which are flanked, so where you have around these variable regions, you have very highly conserved regions where you can put your primers, but will enable to make the PCR. So this is a good point. And the other good point is that this kind of thing is, in fact, you have plenty in the cell, and the number of 18S copies in the genome is very large. It's necessary to have such a large number of copies in order to be able to produce a large number of ribosomes. And so it's easy to sequence. So these are the reference libraries, which are very important if you want to work on 18S. So Silva. So if you Google Silva, you will maybe you add DNA or something like that. But if you put Silva in Google, you will find quickly the website. So it's a very good database. And PR2, which is dedicated to protists. So if you're working on small organism, it's important to know this reference database, PR2. And so here again, and it's a database which is comment dire, the colleagues of Brest no, sorry, Roscoff in the French Brittany, they maintain this reference database. Now let's have a look to the chloroplast. So here you have a scheme of what is a chloroplast. So you see here a double membrane, and here the thylakoids here. And you have here, I think it's, what is it? You have a ribosome here. The small dots are the ribosome, and you have a genome. So you have in a chloroplast, you can have several genomes. And we are going to have a look to these genomes. Here, this is a genome of a particular diatom, Thalassiosira oceanica. So it has been sequenced. It's a circular genome. All these different colors are in fact different genes. And there is one which is important. Where is it? This one here. And we will talk quite a lot about this gene, which is called RBCL. It's a coding. It's coding the large subunit of the ribisco, which is a key enzyme of the photosynthesis. And you will see that this is the really here that we found a good barcode to identify our diatoms. And the reference database that I recommend you is our reference database. No, but I mean, it's in fact, I will explain you tomorrow. This reference database is really dedicated to diatoms and RBCL. And we put a lot of effort to harmonize the taxonomy, to curate it. We were not alone in the lab to do that. I will explain that there are other colleagues that helped us a lot. And so it's an important reference database if you want to identify precisely your diatom species. And you can also use the PhyTool. So PhyTool, this reference database that Benjamin will talk about tomorrow, which is dedicated to phytoplankton. But in fact, we also put all the diet barcode data in this reference database. And you will see it's proposing a lot of different tools that are very easy to, for instance, to see where is the diversity of sequences that we can have. Also, well, so Benjamin will talk about that tomorrow. Another important organelles in diatoms and in every eukaryote are mitochondria. Here you can find a scheme of a mitochondria. So you can see its membrane. So it's outer membrane, inner membrane, and you have some genomes here that are symbolized by these circles. So you have some you can have different copies of a genome in a mitochondria. And here, this is a mitochondrial genome. And there is again here an important marker for barcoding, COX-1. Where is it? It's here. So Agnès told you that COX-1 is a very important barcode, which was proposed as a standard to identify all organisms, in particular insects. But we will see that the story has been a little bit different for diatoms. So just to remember, yes, COX-1 encodes a subunit of a cytochrome oxidase, which is involved in the respiration process, and it's a key enzyme in the aerobic respiration. Okay. Really, if you want to work with COX-1 on let's say any organism, really you have to go to the BOLD system, which is really the Canadian, but let's say a really international initiative. And so there is a huge database here where people put their barcodes, and for COX-1 it's really rich. So if you go to explore the data, yes, you, you click on that. For instance, here maybe it's too small, but I started to write a Bacillariophyta. When you go here, you put search, and you will find here, for instance, all the diatoms that have been sequenced. So it's a very interesting database because, of course, no, it's not of course, but you have a species distribution. For instance, if you click on a particular species, let's say Acnanthidium, you will see here on this worldwide map where there are some samples with Acnanthidium. What else here? Yes, it was just to show you that. So in yellow, you can see that we have some COX-1. And if you click, for instance, on a species, you can have an access to the photos. So here, these are the photos of Irena Kaczmarska from Canada. And yeah, so just to tell you that if you want to work on COX-1, go on BOLD. Some questions about this first part? Or is it easy going? Okay, so let's continue about taxonomy. Just to show you, maybe you know this tree from Adel, where they make a revision of the eukaryotes. So where are diatoms? I don't remember. Yeah. Oh, okay. Just I put some stars, different stars here. Green stars is to show you that yes, we have algae everywhere. So this is explained by endosymbiosis. It's a completely polyphyletic group. All diatoms here are here in the Straminophylla. So in fact, in this paper, which is a very important paper for the revision of eukaryotes classification, diatom classification has been completely revised by David Mann, but I won't present it, this revision here. I just would like to show you the classical taxonomy, just for beginners, if there are some about diatoms. Classically, there are 4 classes for diatoms that are presented. These 2 are what we call the centrics, so Cocinodiscophyceae, Mediophyceae, and here Fragilariophyceae and Bacillariophyceae are pinnate, and here Raphid pinnate. So just to show you with pictures, sometimes it's better. Cocinodiscophyceae, for instance, you can find some filament like Olacosera filamentus. You can see these nice shapes here. So just to show you here, maybe here you have one cell. You can recognize here one valve, here a second valve, and here you have the gridal bands which are connecting the two valves here. And this is a second cell, and it's making some filaments. Here you can find again there is one cell which is under division. You can see here maybe it's a little bit far for those who are in the back of the room, but here there is the old skeleton, so the old valve of a former cell, and here you can see it's under division, and it starts here to produce the new skeleton here. Okay, and sometimes, if you are lucky, you can see here there is a remain of the oxospore here. You remember in the life cycle? So it divides the oxospore, which is here and here, started to divide. Here you have, let's say, the classical cell, and the resting spore is, each part is at the two opposite sides of the filament. And so you have many species in this group. Melosira, I just showed you before. You can see the different chloroplasts. In the cocinodiscophysi, the number of chloroplasts is important in each cell. In the Mediophysi, so another class, for instance, you have a Cyclotella here, many species, very nice. This one is from Lake Geneva, where I'm looking at many phytoplankton samples. This one also, Cyclotella costei. It's another species which is very dominant in our alpine lakes in France. Okay, let's continue. So let's forget, all these species are planktonic. You see they have no raphe, they are centrics. The raphe is this fissure that enable diatoms to move. They don't need to move when they are planktonic. Fragile Ariophysi here. So these are pinnate diatoms, elongated. This one has no raphe again. Okay, you see the different shapes. This one is planktonic, again from Lake Geneva. I can see that in the plankton. Here also, it's a pinnate diatom, which is very common in the plankton of Lake Geneva. Okay, some diatoms, diatoma. You can see the shape is different with long, with these costae, which are going from one side to the other side of a frustule, and making some colonies, zigzag colonies. Here you have one cell here, another cell here connected to another one, and these diatoma have several chloroplasts here. There are numerous chloroplasts. Let's go to the Bacillariophysi. So here you can see we have monoraphids. So what are monoraphids? So we still have our two valves, one valve here, another one here. You can see, so here it's, let's say, a silhouette. Do you understand silhouette? I don't know the English name. Okay. And here it's the phase view. And you can see again these two valves, and only one of both has a raphe here. Here you don't have. That's why we call them monoraphids. And there are many species in the acanthidiums. Here you have two species. Minutissimum is a very common species in rivers. Catenatum, you can find them in lakes. Biazoletianum pyreneicum, here you can see it's again a monoraphid. You can see it's two valves. What else you can see here? Now we are in the biraphids. It means that the two valves have a raphe. Some are heteropolar, like this big Gomphoneis, very nice. Some are curved with this raphe, which is, you can see it. And some sigmoid. This one is living mostly in sediments. Some are straight. Pinularia living in acidic waters, and so on and so on. So you can see that each species have a very particular ecological niche. Okay, we have also these diatoms who have a special raphe, which is called a canal raphe here. But you see that all these diatoms have a very particular niche, and this is why we use them for biomonitoring, because we can use this diversity and the specific ecology really to assess with precision in our case, the pollution. Some questions about diatom taxonomy? Maybe you're not here for that. Okay, so let's go to classical diatom biomonitoring. So what is the usual methodology for diatom biomonitoring? So there is a standardized methodology, which is based on microscopy. So there is a European standard that I think most of you are using, even in Serbia and everywhere. We are using, everybody is using the same methodology. So what we are doing when we go into a river, we are sampling stones, like you can see here, and we are brushing the biofilm with a small toothbrush to detach the biofilm, collect it, and then we put it in a small vial. We put formaldehyde or ethanol. It's better to put ethanol if you want to do metabarcoding. But here I'm going to talk about the classical methodology based on microscopy. When you're back to the lab, what we are doing is a sample treatment based on mineralization, because the identification of diatom is based on the frustule, and so we don't need all the organics and so on. So we remove all the organic matter and the intracellular organics which are inside the frustule. So we remove that, and we make a slide preparation. We look at that under microscope. You see many different shapes. All these shapes, you have to give them a name. You have to count 400 fustules. So all these fustules, you have to count them and say, okay, we have species A. How do you do that? In fact, you need a large library with many books. And so you open these books. In this book, what do you see? You have pictures. And so you compare the picture you have in the book with what you see under the microscope, and you make a relation, and then you give it a name. And for this, you need a lot of memory, good eyes, and you play the game. In the end, we get a species inventory. So with species A, 300 frustule, species B, 90 frustule, and species C, 10 frustule. And with the species inventory, we calculate a biotic index. And this biotic index is a way to assess the quality of the environment, to assess the pollution of a river. What are the principles of a biotic index? Most of the diatom indices, let's say most of the biotic indices are based on the formula of Zelinka-Marvan. So you can see it's not, it's quite old, but it's still working very well. It's based, in fact, on ecological profiles, which are weighted by the abundance of the species in a sample. What is an ecological profile? So we have a graph here. So you can see here on the horizontal axis, you will have a nutrient level, and here the presence probability of the species. So you can see here that this species will be dominant, will have a high probability of presence in low nutrient level. So it's an indicator of good water quality. And you can measure two different things. So the first thing is, let's say this peak here, it will be the indicator value. So the indicator value tells you that this species is a good water quality, has a good indicator value because the nutrient level is low. And the stenosis degree. So the stenosis degree is measuring, in fact, the width of ecological profile. So if it's restricted, the stenosis degree will give you a high weight to the species. It will tell you that the species is very indicative. And on the contrary, if you have something which is relatively flat, in fact, the species won't be very indicative of any nutrient level. Let's take an example with three species, Anosia intermedia, Nitzschia fonticola, and Mayamaya permitis. So usually, are there some people working with diatoms here and counting diatoms? Please raise your hands. Okay, many people. How many species can you find in a sample usually? Yeah, yeah, but allez, let's say approximately, not three. Do you agree? So here it's really a stupid example. Usually we don't have three species, but it's mostly 10, 20, and even more species. So let's imagine we have a very very low diversity sample with media usually is a good water quality indicator. And it will have this straight ecological profile. Niche fonticola, well, it's not the reality. I know that there are very good diatomees, but it's a species where the ecological profile is quite flat. So this species won't be very indicator. So you will give a quite low power, a low weight in the calculation. And Maya permittis indicator of bad water quality. Usually you can find it downstream wastewater treatment plants. So you counted and you found 90% of this species, 8% of this one, and 2% of this one. And here is it's in fact a simple weighted average of this formula, Zelinka and Marvan. It's a weighted average where you have A, this is the abundance of a species. So if we take for Eunosia intermedia 90%, that you multiply by it's the indicator value. So here it will be a good indicator value. And you multiply by S, so the stenoicity degree. So here it will be a high a high weight that you give to the species. And you divide by its abundance and its indicator value. So basically, most of the biotic indices are like that. What kind of Diatom index are you using in Serbia? IPS, yeah. I think it's based on this formula. You agree? Yeah. So in France, we were using the IBD, Indice biologique diatomé, which was an index developed in the framework of the WFD, so the Water Framework Directive. And there was the first publication in 2000, and it has also been standardized in 2000 at the French level. So it has been constructed on a database that you can see here, some samples which were in fact collected by Michel Coste, 1,300 diatom samples where you have floristic list, but also chemical parameters to be able to calculate these ecological profiles. So it was the first version of the Diatom Index IBD. Then we had a second version because we had this Water Framework Directive, which asked us to assess the pollution of the different rivers in France. So it was a way to recalculate the ecological profiles of the IBD. So you can see almost 3,000 Diatom samples where you had floristic list and chemical parameters, and they were able to recalculate the ecological profile of each Diatom species. And same story, so it was 4 years ago, this time with 5,000 Diatom samples. So now we have really a robust index, which is really, yes, robust, at least for French rivers. How IBD is used to assess the river quality, so the river ecological status. So the river status is based on 3 quality parameters, the biological quality, and in the biological quality, so you find again our diatoms, but also macroinvertebrates, fish, and macrophytes. The second parameter is the physicochemical quality, and the third is the hydromorphological quality. These 3 quality parameters are there are rules to organize all these 3 parameters to give a quotation that you can see here. I won't enter into the detail here because it's not the topic, but just for you to know that there are these 3 kinds of parameters and there are some rules to say, okay, if we have this quality for this ecology element, so we have this quality in the river, but it's not the topic. This is the first map of France with all the quality with the different ecological status. What you can see here, we have different islands. Guadeloupe and Martinique, in fact, they are in the West Indies near Cuba. You see this area. This is French Guiana, so in South America. Mayotte, it's an island which is near Madagascar, and Réunion, it's in the middle of Indian Ocean. So since they are part of Europe, we also have to assess the quality for these lakes, for these rivers. Yeah. And this was in 2019. You can find here also the mainland France and the different islands here that I told you. But yes, I just would like to come back to 2009, and you can see here this island, which is called Mayotte here. Nothing. There was nothing. No evaluation at that time. And in the meantime, we had the first PhD student, so Lena Kermarek, and she already made the proof of concept with DNA metabarcoding and diatoms, and she showed that it was possible to identify diatoms with DNA metabarcoding. We presented in the meantime this result to water managers, and they were very interested in these results. And so they asked us to propose biomonitoring tools for Mayotte, so classical one based on microscopy for diatoms, for macroinvertebrates, but also to test DNA in this new island, in this island where there was nothing. In fact, just to explain you, Mayotte was before, I think it was 2011. It was, let's say, a territory, so French territory, and there was a referendum, and people voted to stay in France. And so it became officially completely French. So it's a French department as Haute-Savoie, where we live. And so it's fully part of Europe, and we had to apply the WFD there. And this is why the water managers proposed us to test DNA metabarcoding on this small island. And yes, the objective, so we will talk about this example because this is where we have our experience to develop Diatom DNA metabarcoding. It's really our laboratory where we tested all these things. And so really, just to make you understand what we're going to show you in the next presentations, the idea was really to replace the microscope determination with DNA metabarcoding. So to change from this to what showed you Clarisse just before. Okay, do you have questions? No, no questions. Oh, just 5 minutes late. It's okay. So no question. Thank you.